Hi, I'm Chance.
And I'm Sarah Catherine.
And this is Conservation Connection.
Presented by Last Chance Endeavors.
We run a wildlife education nonprofit focused on connecting students to their environment.
Each week here on Conservation Connection, we do just that by introducing you to the groundbreaking science and conservation work that's happening every day around the country.
We talk to professionals in the world of conservation science and wildlife management and ask them about their career,
their current projects,
their wild and crazy stories from the field,
and everything in between.
Join us each week to discover just how these dedicated people are working to protect our planet. Alrighty guys, welcome to another episode of Conservation Connection. We are so excited to be back here at Grice Marine Lab for their research experience for undergraduates in 2021. This is an amazing program, and you guys are about to hear from four really cool researchers, undergraduate researchers who are doing neat things here in Charleston this summer. So we are here in the room with Regan, Zoe, Joelle, and Charles, and we're going to let them tell you a little bit, just 10 seconds, about their projects.
Welcome to the show, guys.
Thanks.
Just real quick, what are you doing here in Charleston this summer?
I'm looking at the sediment chemistry of the Spartina marshes and the surrounding areas in Charleston, and I'm also looking at the marsh profile.
Awesome. And Joelle?
So this project is actually a little different. I'm studying biosignature volatiles of extremophilic archaea. So I actually study more astrobiology related stuff, which is pretty cool, I think.
Pretty fun in a marine biology program.
Yeah, and it's really cool learning more about marine biology because I didn't realize how amazing that field was. Some really cool stuff going on.
Absolutely. Zoe, what about you? What are you doing this summer?
I am looking at the possible varying effects of ocean acidification and increased carbon dioxide on crab performance post-molt.
All right. And Regan?
I am looking at the long-term impacts of salinity exposure in amphibians in the local area.
All right. This is the whole smorgasbord of projects here. I'm going to go ahead and start by diving in with the crabs. So you're looking at how carbon dioxide levels are affecting crabs after they molt. So in what way does carbon dioxide have an effect on crabs? Why would that be an issue for crabs who are going through... Well, actually, let's start here. What is molting in crabs?
Okay, yeah, you're right. There's a couple different layers to this explanation. So molting, we'll start with that, because crabs are organisms that have... They're not like me and you, where we have our skeletons on the inside and we have our skin and our flesh on the outside. Crabs have all of their internal organs and their flesh on the inside and then are covered by a hard exoskeleton shell. But because of the rigidness, the hardness of this shell, as they grow, as their internal body, their muscles, their internal organs, as they grow, they tend to kind of squeeze this shell and it doesn't really form with them. So this requires the molting of this shell, or the getting rid of this shell. And then as they do this, their soft body is exposed, and so they use minerals from the environment and they regrow this calcium carbonate based shell.
So basically it's like wearing a sweater and you outgrow it and you have to get that sweater off and then remineralize a new, bigger sweater on your body.
Right, and that new one fits them better. And as this happens, there are several things going on with their internal organs as well as the tissues and the muscle connections that connect to this exoskeleton. They have to first detach from this shell to get rid of it, and then as they regrow the new one, all of these new attachments have to kind of take place. And so that's why I'm interested in the performance of these crabs and the effects that carbon dioxide will have on them, because this requires a lot of kind of musculoskeletal interactions. So rather than looking right at their shell and their recalcification, I'm looking at the muscle interactions and how that's going to affect their strength and their functioning as a crab.
I can't even begin to think of how I would measure the musculoskeletal interactions in a freshly molted crab. So what methods are you using to sort of determine their performance in varying levels of carbon dioxide, which we will get to in a second about the carbon dioxide.
Right. Like I said, the multiple layers. So we are looking at two different methods right now, one of those being their pinching force. So I have a sensor. It's think a long piece of plastic with a little circle at the end, and the circle at the end is a little pad. And so I pick up the crabs and I kind of like agitate their claws a little bit and get them to pinch on to this pad. And the pad reads this kind of compression, converts it to a force measurement, and we are measuring in newtons. And that kind of connects to the software that I have on my laptop, and it shows a force reading for the pinch that they're doing. So that's kind of like a measure of their strength, I guess. And then the second that we're looking at is the cuticle regrowth, because there's a hard shell, right? And then the muscles are connecting to it. And so as muscles compress, you have to look at if the shell is soft, the compression of the muscles will kind of like bring that shell to it or bend the shell a little bit, versus if the shell is really hard, then the compression of these muscles won't necessarily have as much of an effect on the hard shell. So we're also looking at the physical hardness of this shell we're using a force gauge, and it kind of can measure compression or tension. We're obviously doing compression, and as the gauge comes down, it measures the distance that it's coming down, and when it presses down on the claw of the crab, it will measure the distance versus the force that's being put upon it. And so we're not crushing the shell, but we're kind of looking at how much that shell will compress when we press down on it.
It's a very scientific way of saying that you squeeze it and see how far it bends with a certain amount of pressure.
Exactly, right.
Yeah, absolutely. And I feel like that's very important, because molting represents the most defenseless time in a crab's life, right? They don't have their suit of armor. They're totally naked. And so how quickly their defenses come back online is really a very important measure of, are they going to die or are they going to live?
Right.
With all of that said, why are you looking at how carbon dioxide affects these things?
So this kind of goes back to what we always hear about greenhouse gases getting trapped in the atmosphere. And carbon dioxide is one of these gases. As the concentration of carbon dioxide increases in the atmosphere, that means more carbon dioxide is getting absorbed into the ocean. The process of carbon dioxide mixing with water and getting absorbed into water creates carbonic acid. Carbonic acid increases the acidity of the water. So with that is the process of ocean acidification.
Right. So think about how a soda is bad for your teeth, not just because it's sugary, but also because it's really acidic, because it's carbonated, right? So that CO2 dissolved in the soda itself is creating that carbonic acid, which, basically melts your teeth.
Right. And so with increased acidity in the water, that affects the compounds in the water, such as calcium carbonate. And calcium carbonate is what a lot of these calcifying organisms, think crabs, anything with a shell, corals even, which is why we hear a lot about corals, coral health declining.
Don't forget algae.
Right.
Said the man studying Spartina.
Can't forget about those algae. So this increased acidification is going to potentially cause problems for organisms who require this deposition of the calcium carbonate to undergo their various processes and life functions, because if there are varying levels, that affects how well they are able to use that mineral and thus live their life.
And survive.
Yes.
Right, exactly.
Right,
yeah.
right.
So it basically boils down to that the more acidic the water is, the harder it is to pull calcium carbonate out of solution and into a shell.
Right. That is the idea behind what I'm studying, at least.
Right. So we're going to get to results in a minute. But Charles, I want to hear a little bit more about how you're studying the pluff mud, which for those of you who don't know what pluff mud is, Charles is about to tell you, and the ways that it's affecting Spartina grass.
So pluff mud is this lovely mud in the Charleston area that gives its, Charleston area the smell. It's essentially the mud that's around the area. What I'm looking at is the pluff mud in the Spartina marshes. And so Spartina marshes, or the new name is now Spirobolus alterniflorus. The Spartina marshes, surround the entire area. If you're ever going to the beach down here in either Folly Beach or Sullivan's Island, you're going to see all these like marsh areas. That's all Spartina.
And it's the grass, right? That's the dominant organism and is like the stabilizing force in these mud marshes.
Exactly. And you can kind of tell that if the Spartina weren't there, the other organisms that also live there, like crabs, snails, fish, all that sort of stuff, they're not going to also live there. And the Spartina, the productivity of the Spartina is indicated on the healthiness of the soil. And so it's kind of like if you have your own garden at home, your tomatoes aren't going to grow well if you don't have good soil, right? And so that's why you use nutrients and you put in phosphate and all this other stuff. And so what I'm looking at is how healthy that soil is and the different properties that this soil has. Specifically, I'm looking at these two different groups of Spartina. One is creekside, where the Spartina really grows tall, about like maybe my height or taller.
And he's a tall guy. So
I'm about 6 foot. So
tall grass.
Yeah, very tall grass. And then the other side of the coin is the short grass, which is the more inland grass, and it's a little bit higher up on the marsh. And so I'm looking at those two extremes just to see, is there a difference in these soils, and can we indicate if there's going to be a difference in the soil? Is there going to also be a difference in the Spartina, the actual plant that's growing in it?
When you're looking at the soils, what sort of parameters are you checking? Are you looking at like mechanical parameters? Are you looking specifically at chemical parameters? What are you doing?
So I'm looking at a little bit of both. For example, for a little bit of like a physical property, I'm looking at the actual particle size of the mud that's there. And so if you go to a bunch of different marshes, some of the marshes might be sandy, some of the marshes might be a little bit muddier. And so I'm actually using a little bit of a sample from those marshes, and I'm using this big machine with this big laser, and I'm actually able to measure the particle size of the mud that I put in to the machine. And so that's something that's really cool.
That's one of the more sciencey things I think I've ever heard.
Yeah, it's literally like a laser and then a camera, and it's like, oh, this is how big it is. And it goes down to about like some sort of like pictometer or something, like really
super tiny.
Yeah, 10 to the minus 9 or something, like crazy small. I just finished all that data and I have to process it now. So it's,
The other side of doing really fun science things is looking at data for hours and hours and hours.
Oh yes, it's great. And then on the chemical side of things, unfortunately I don't have, enough money. I would love to look at literally every single parameter that I can, but I'm looking at ammonia concentrations. And if you don't know, ammonia is one of the key nutrients for, plants and algae, basically. So I'm looking at the concentration of ammonia basically to tell, is the soil good? And so that's one of the other chemical properties that I'm also looking at.
All right.
What made you want to study this? Like, why is this your research project?
Yeah, you might ask me, why am I trancing around in the mud, covered in mud or whatever? No, so this entire last year, my mentor kind of got in contact with him, and I guess this is going a little bit more into like how you get into this sort of thing. But I contacted him and I emailed him because I read one of his papers on this invasive species called Garaphyton, and the paper called it Gracilaria. I think you guys...
I know it as Gracilaria, yeah.
Yeah, and it's Eric Sadka. And so I emailed him and I said, hey, what are you doing? And so last August, he's like, hey, why don't you come in? We can talk about, what I'm doing right now. And he said, I'm working with Spartina. And he said that he was specifically working on these two different zones of Spartina, the tall and the short. And so I was like, oh, that sounds cool. So we just started emailing back and forth, and I started reading a bunch of papers on the topic. And then I was like, hey, I think, this is something interesting I want to do. What do you think a possible project that we could possibly work on? And he said, we could do this. I'm like, I'm more of a chemist now. I've kind of developed a, chemistry background. And so I asked, is there any sort of, chemical properties that we can investigate? He's like, yeah, no, we could look at the soils. It has been done in a wide range of marshes across like the East Coast, but it's never been done in Charleston specifically. And one of the papers that I was reading about the marsh sediment in, I think it was in North Carolina or Virginia, they were saying that there was like these different levels of nutrients because of the way the marsh looked. And what I mean by that is instead of the marsh just going up in elevation as it goes, so tall to short, it's tall is low, short is high. They said that tall was high and short was low. So that kind of makes a huge difference when you're asking about the salinity, the nutrients, the sediment type, the water, all this other stuff. And so eventually he said, hey, why don't you come over here to Fort Johnson and talk to Bob and see if you can get some money for the summer? And I said, okay. And I applied, and
here we are.
And now we're here.
You're out in the marsh fighting pluff mud.
Yeah, no, it's been great. So pretty awesome.
Excellent.
Yeah, and for anyone listening, we did do an episode with Charles's mentor, Eric Sotka. It's a really cool episode, so...
Yeah, check out the show notes. I'll put the link there. It's in season 1, so if for some reason that link doesn't work, just scroll down. All right, so I want to hear, we're going to go next to tadpoles and salinity. So first off, do tadpoles care about salinity?
Yes, they do. For those of you who are not amphibian experts like myself, amphibians are not marine organisms. They have to have completely fresh water. They don't like salt at all. And so what's going on is a lot of the freshwater sources on the coast and in the north are getting saltier. A lot of times in the southeast on the coast, we have these big hurricanes now with climate change and things happening. We have these huge storm surges. And if you live in Charleston, you know the city is below sea level, lots of flooding. And so all this ocean water is getting into our freshwater sources and making them salty. And in the north, Charles is from New Jersey. I'm sure he could tell us all about it. They use salt on the streets to prevent ice, and all that salt runs off into the ditches where the rainwater is stored, and that's where the frogs like to hang out and lay eggs. But now all of a sudden, this fresh water is being salty, and that's a problem for them.
How salty is salty?
That's a great question.
From pristine stream water to ocean water, how salty is a problem for frogs?
That depends on the species. They have found that it is variant among species. So for reference, ocean water is around, we'll say 28 parts per thousand salt. So for every 1,000 water molecules, there are 28 molecules of salt. That's ocean water. There's been research studied that because of flooding, freshwater ponds can get up to about 9 parts per thousand salt.
Okay.
My tadpoles struggled with 6.
So it's really like very low levels comparatively of salt are having big problems.
Because amphibians are super cool. They have to grow up in the water, right? When they're eggs and then they become tadpoles, they have to have water. And then they go through this metamorphosis and they grow legs, which is fun. And then they can go on land and they can hang out on land then. But something cool about amphibians is they have this permeable skin. So they breathe through their skin, they drink water through their skin, they interact with their environment through their skin. And so if they're hanging out in these little pools of water that all of a sudden now have salt, all the salt is going to cause the water to escape their bodies to try and decrease the salinity of the pool instead of the water hydrating them.
Right, that's osmosis. That's just water moving from one pressure to another.
Exactly.
Right? It's the same issue that's why salt kills slugs, right? Because
It's
the salt
100%.
just pulls the water right out of their bodies.
It's the same thing.
Right. So unlike mammals, humans, us, we have this lovely skin that has adapted to put a really strong barrier between us and our environment because we live and die on land. So we need to keep all of our water inside our bodies, and that's what our skin is adapted to do. Frogs and other amphibians, on the other hand, don't have that kind of protection because they live in the water. Why would they need skin that's adapted to retain water within their bodies when they live in the water? So the mucous membranes that they have on their body is similar to what we have like inside our lips or our mouths. So imagine if you spent your life walking around and your whole body was just like the inside of your cheek.
That's terrifying.
Right? Exactly.
That's just a terrifying image.
And then these giant aliens start dumping salt everywhere because they don't want to slip off the roads. And now that's a you problem, right? So that's kind of what this issue is. So how are you studying the effects of salinity in amphibians?
I stole toads from the wild.
Collected. She collected toads.
Kidnapped.
Well, that's kind of funny. Dr. Welch, my lab mentor, and the other student in my lab Aubrey Anthony would walk around at night and like sneak up behind the frogs and then like strike their hand out and catch them. And I was like walking around following toads, being like, come here, please, come here. So I don't know if I really collected them, but I caught some. I caught some toads and I brought them back into the lab. And this is their mating season. So they were looking, to get partnered up. And so we brought a total of 7 pairs of toads back into the lab, and we had these huge containers filled with like a control solution that they can be comfortable in. We gave them some time, let them do their thing, and they laid eggs, thankfully. Toad eggs are really cool. I'm working with the southern toad, or Anaxaris terrestris, and these specific eggs, they're little black circles coated in a clear jelly, and they come out in strands, and it's the weirdest like spaghetti noodle kind of thing you'll ever see in your life. But I collected, we probably had in total somewhere around 10,000 eggs. Which
from
was
7 pairs.
Right, so one pair can have anywhere from 2,500 to 4,000 eggs in one clutch, which is just one like set of eggs, yes. So yeah, all of a sudden I became a mother of 10,000 and I was not ready. But that's okay. I took sections of eggs and I confirmed their fertilization. Like, okay, these ones are going to make it. They're going to grow. And I put them in different salinities. I had a bunch of controls. So we keep them around at 0.4 ppt, or parts per thousand salinity, which is about what you would find in like a natural freshwater pond. And then we had 4 ppt and 6 ppt. I raised them from little eggs to hatchlings to tadpoles. And when they got big enough, I moved them into their own rooms, their own little containers with the same solution. Some of them, it's a little complicated. I took some of the controls, so they grew up in like fresh water, and I moved them to salt water. Like for them, all of a sudden there was a hurricane and now there's a bunch of ocean water in their home. Some of them I took out of salt water and I put them in control. So that's like if it rained and a bunch of fresh water all of a sudden filled up their little pond and it's not as salty anymore. And some of them I just continued in the same solution just to see if they are able to escape this dangerous salty solution, would they make it out okay?
So, assuming that we have already released some of our other REU episodes and that our listeners have listened to them, how is your research different from Aubrey's research on tadpoles?
Right. Aubrey is looking at a different species, which is completely insane. His Hylacenaria can survive all the way up to 8 ppt, which is really nothing, but it's a lot.
It's a lot for a frog.
Well, I don't know if you want to keep this in. All of my 6 PPTs died, which was sad times for me. I lost about 600 kids.
At the same time.
Yeah, it was a lot. But, we move on with our lives.
It was a rough week. Lots of ice cream.
But Aubrey is looking at how necessarily the salinity is going to affect the tadpole's relationship with another organism, with their predator. I am looking at the development of the tadpoles, of the organisms themselves, and I am looking at if we are able to take them out of this dangerous environment, are they able to survive as they would have if they weren't in it at all? So essentially, I'm looking to see if they can recover from their childhood trauma.
So you're just looking at straight mortality rates post-salinity exposure?
I'm looking at mortality rates and growth. We're weighing them, which is fun because some of them are getting like really big and I'm proud of them. And once they metamorphose into toads, we will be looking at performance like Zoe is. We'll be looking at like how far they can jump. Can they jump for long periods of time?
So you're looking at a whole suite of like physiological and developmental questions.
Yes.
Okay. That is a bear to tackle in a summer-long project.
Yeah, especially because toads take about 1 to 2 months to metamorphose. So, it's definitely going to extend a little bit into the fall.
So you're on toad time.
Oh, 100%. All the time. It never stops.
All time is toad time.
Yes.
Never
100%.
stops.
That's awesome. So moving on to our last person in the room, Joelle, studying archaea. Yeah, just jump in and explain to us simply what you're doing.
Hi. Okay, so it's a little bit complicated. That's what I've learned. I tried to explain it to my 85-year-old grandpa, and it took like half an hour. So I think I have it like pretty down pat after that.
Good.
So the title of my project is Biosignature of Volatiles of Extremophilic Archaea. A lot of jargon, doesn't make much sense to even me when I first read it. I was like, oh, this is what we're doing. So essentially what I do is I look at other planets and I think, oh, well, if we have life here, what if we had life over there? Classic astrobiology stuff. If we had life over there, what would it look like? So think about Mars. I think at this point we kind of all know Mars has water on it, just like Earth, but a lot of it's evaporated. So what happens when you evaporate water? Salt water, specifically. It gets very salty. And I'm kind of doing the opposite to Regan. I'm looking at organisms, archaea. So for people who don't know, it's kind of off track, but archaea, just so I can...
Yeah, what is archaea?
So archaea, there are three domains of life. Bacteria, archaea, and eukarya. So eukarya is what we are. They're multicellular organisms. All animals, plants, stuff like that, they're all eukarya. And then there's bacteria, which I'm sure we all probably know what that is. Single cellular, classic, gross stuff. And then there's archaea, which have only really been around or categorized since 1977. So pretty new stuff in the scientific world. A lot of people haven't really done research on it. And so archaea are also single cellular. So those are called prokaryotes. So bacteria and archaea are both prokaryotic. But archaea are more similar to eukarya in the fact that they have what's called histones. So when you think about DNA, bacteria and eukarya have very different DNA, essentially same building blocks, but we have about 2 inches of DNA in a chromosome. And our cells are very small. So how do you compact that? Well, for eukarya and archaea, this is what makes archaea special in one of their ways, they have proteins called histones. And that is what the DNA is wrapped around very, very tightly that allows us to have so much DNA compressed into such a tiny space.
So basically, they're the toilet paper rolls of DNA.
Exactly.
So it wraps around that so that you can have a long length of DNA in a very small space, just like you wrap toilet paper around the roll so that you can have a lot of it in a small area.
Exactly. So that's why archaea aren't bacteria, one of the reasons. Another one is their cell membrane. They have different components than bacteria and eukarya. So archaea are kind of this weird mixture. I like to consider it like the missing link, even though it's not, between bacteria and eukarya. So I'm looking at archaea because one of the fun things about them is they love crazy environments.
We call them extremophiles, which literally means extreme lovers.
Exactly. So these archaea that I'm studying, looking at Regan's toads, it's a little different. My archaea love salty water. And we're not talking about ocean water. We're talking about like Dead Sea water, Great Salt Lake water. I work in 23 to 28% salt water.
Is that by weight or by volume?
By volume.
Okay, so that's, I mean, either one is a lot, but that's a lot.
It's a lot. So they're called halophiles, which means halo is a term for salt. Philes, like we said with extremophiles, love salt. So these halophiles love salt. So these are the ones that we're looking at for Mars, and we're also looking at Venus. So not necessarily the surface of Venus, because nothing can grow on the surface of Venus. It's way too hot. You would die immediately. But we're looking at the clouds, because prokaryotes can survive in clouds because they're very small and they're cute.
And we all know that cuteness is like the defining factor for survivability in clouds.
It is. I hope if we
Yes.
find aliens, they're cute. I think that's like my biggest hope. So we're looking at the clouds of Venus, and if anyone knows anything about Venus, it's very hot, even in the clouds, and very acidic. So the archaea that I'm looking for those are called thermophiles. They love hot places. Currently one that I'm growing called Thermoplasma acidophyllum. Its optimum growth temperature is about 60 degrees Celsius.
So what is that in Fahrenheit?
So that's actually 140 degrees Fahrenheit. So we're talking about like when I dry flowers, that's about the temperature I dry flowers at. So this is like some serious heat. Like we have like a little oven, and I like check my archaea in the oven, see how they're cooking, stuff like that. And then,
they have to be in that oven to survive.
Exactly.
That warm temperature is what they need to be able to reproduce.
Yeah, they won't grow unless they're in hot temperatures. And going off what they need to survive, they're also acidophiles, so they love low pH. So I know with Zoe, we were talking about acidification of oceans. And once again, extremes, they grow in pH 0 to 3 with an optimum pH of 1.5.
And water that is perfectly neutral is 7.
Yeah.
And it's a logarithmic scale.
Yeah, so we're talking about like battery acid.
Yes.
Like I had to do full PPE, like coat, gloves, eye protection, hood fume. It was
Like,
serious stuff.
Blindness would destroy the inside of your lungs if you breathe the fumes. Like bad.
Yeah, so it's pretty serious stuff. So
And it's the perfect nursery for this bacteria.
they love it. They love it. They thrive in it. So that's the type of stuff I'm working with. So what I'm doing, my project, so that's just an introduction on the cute organisms I work with. Not as cute as frogs or crabs, or Spartana, but still pretty cute. So what I do is, we aren't on Mars, in case you haven't noticed. What do we do when we can't see something? Well, we can smell it. So essentially, I'm starting to smell this archaea with what they produce. So when we think about it, we produce things. We fart, we burp, I know, little bit of potty talk, but it smells really bad, and you can smell it. Archaea do the same thing. They produce what we call VOCs, which are volatile organic compounds. So these are compounds that have carbon in them and are also gas at room temperature.
Which is what volatile means. Like a lot of times we hear volatile and we're like, oh, it's going to explode. And the reason that volatile things explode is because they're gases which can ignite easily. So volatile literally just means at room temperature, this goes into a gaseous form.
Exactly. So we would be able to tell if these were in the atmosphere. So what I'm looking at is I'm growing these archaea and I'm saying, well, what do they produce? What volatiles can we see when they grow? And if we see volatiles, we send that over to NASA, our friends at NASA, and we say, hey, we found these compounds, and hopefully they'll be pretty complex. So like if we find methane and stuff, methane comes from geothermal activity as well. So it'd be hard to tell, is this coming from the planet itself or life? So if we find like complex, long, crazy molecules, it'll be a little bit more certain. So we send it over to NASA and we say, hey, did you find any of these in Mars's atmosphere? And if they say, yeah, and we don't really know where they're coming from, we can say, well, we know where they're coming from. That is life. So essentially that's my project.
That's life, NASA.
Yeah.
So really what it boils down to is that these archaea create compounds just in the process of living for them. And those compounds are signatures. They are only, hopefully, created by life that is doing similar things to these organisms. And so if we find those in places that we don't expect life, it's a good sign that there may be some form of single cellular life on that planet.
Exactly, yeah. And we'll be able to tell, hopefully, what kind of organism it is based on their VOCs, because the other part of my project is biosignature, like you said. It's like a signature for the organism. They create different quantities, different compounds based on what they are. So if we can match it up, we can say, oh, this is thermoplasma acidophyllum, or this is Halopharax falconi. Like, we'll be able to tell, hopefully, what species it is, or as close to what it is as possible.
Right. It would be earth-shattering if we found a species that is present both on Earth and on Mars, like identical to each other, because that would imply that at some point life traveled from one planet to another and survived.
Exactly, yeah. To survive the vacuum of space, because Earth is constantly throwing out rocks, and we get stuff from Mars, and Mars gets stuff from us.
We're like little pen pals.
We are like little pen pals. It's pretty interesting. I saw like a rock from Mars in one of my classes, and it was like the coolest thing ever because it's from another planet. But we could technically have sent organisms to Mars, but they probably wouldn't survive because it's a barren wasteland.
Unless they're like some sort of weird organism that, really likes extreme environments, like, say the vacuum of space or extremely salty water.
Exactly. Unfortunately, my organisms are aerobic, so they do need oxygen, which there's oxygen on Mars, but unfortunately there's no oxygen in space.
Right,
So
yeah.
I don't know if they'd be able to survive the trip over. So maybe not great pen pals.
Yeah. Just can't send living organisms.
Exactly. We can send dead ones. That's fine.
So, what is your long-term goal with this? Like, do you want to keep doing research on this? Do you want to work for NASA? Like, what's up?
So I would love to keep doing research on this. Obviously, like my life goal now is finding aliens. And it's pretty funny. I did not expect to be an astrobiologist. I was actually terrified of aliens as a kid. That was one of my biggest fears growing up. I wouldn't be able to sleep thinking that aliens were going to abduct me. So did not see this career path coming. But
It's a strong passion that happens in weird ways, you know?
it is. It is. So I would love to work for NASA. I would love to keep doing what I'm doing, especially with extremophiles, because I just think they are so cool. And halophiles are pink, which is my favorite color. So I would love to keep working with pink organisms. And yeah, so NASA's the dream. Working at an astrobiology institute for NASA would be insane. I'd also love to teach, because I feel like when we think aliens, we think, oh, little green men, that like Area 51 stuff. But it's so much cooler when you think like such mundane things can be aliens. Like archaea are all over. If you go to the Great Salt Lake in Utah, we have some cultures from there that we use that I'm culturing currently. It's so mundane. You can find aliens basically in your backyard.
In Yellowstone, they have these really hot geothermal pools that are beautiful because they've got all of these extremophile archaea living there and just like totally vibing in this crazy hot pool. They're not uncommon on Earth, and so it's definitely something that we could see somewhere else.
Very cool. We have 4 of y'all here. We could keep talking forever, but I do want to go around and get the answer from the rest of you guys of what are your career goals after this, and either like what's been your favorite experience or recommendations you have for students who want to do something similar.
Yeah, so in the future, I will probably go on to either get my master's in marine chemistry or an oceanography degree, and possibly a PhD in oceanography or marine chemistry. Not really sure. I came to CFC thinking about, I want to do marine biology, and then I took chemistry here. And I just like fell in love, and I absolutely love chemistry now. I know that might be insane for everyone else, like looking at me.
It
like,
really is.
I have like 2 heads.
Actually is.
I took organic chemistry, and I was like, this is what I want to do.
Said no one
right?
ever.
Exactly.
Said no one ever.
So yeah, I found out that I like chemistry a lot, and this project is very chemistry oriented, and so I definitely see myself in the future going into some sort of field where I can do chemistry research. If that's in the next year, maybe. If that's, in the next 2 years, maybe. It's a lot of school, so I have to really, think about it. It's a big decision. But yeah, and what I really like about this experience, REU, is it's kind of like building up connections and building up different, I guess, like a network of people and what people are doing and all these different things at CFC in South Carolina. And, I'm sure you guys have explained before, but this campus right here, Fort Johnson, there's like NOAA, NIST, DNR, and College of Charleston all in the same area. So you get all these connections, all these people you get to meet. And that's one of my favorite parts. Specifically, I really enjoyed, we went down to Ace Basin, and we had like a little experience where we like went out on a boat, and we were looking for like horseshoe crabs, and we like saw like a bunch of turtle eggs and all this sort of stuff. So I really just like getting out in nature and like doing all that sort of stuff. But yeah,
Was there ever a moment when you were just like stuck in the pluff mud and you're like, why did I do this?
yeah. I began like in the beginning of the summer. I was like very nervous to go out in the pluff mud, and I was like, oh God, what if I get stuck out there? I have to bring someone along, blah, blah, blah. Who wants to go along with me? No one wants to like join me in the mud, right? And there was one time where I think it might have been like 3 or 4 weeks ago. It was a Friday afternoon, and I was going to about 6 marshes that day, and I was on my last one, and it was like 3 o'clock in the afternoon. I was drenched in sweat, covered in mud, and I remember just like thinking to myself, oh, this is why I'm out here. Like, this is like, I might be miserable and like disgusting and sweaty and just tired right now, but like, I don't know, I looked out and I saw like a dolphin like pop his head up and I was like, this is awesome. I'm really enjoying this. And I'm like, if I'm that committed to going out to the marsh for that long, I think, I'm pretty much in it for the long run, I guess.
Absolutely.
Yeah, you must like it.
Yeah, I must like it. And then I guess some advice for some incoming students, whether you're in high school or you're in college and you're kind of not really sure what you want to do, you just got to put yourself out there. Biggest advice for a high school student that's thinking about a marine biology degree or anything in the natural sciences, either chemistry, geology, or just biology in general, get yourself out there. Try to experience as much as you can before you go to school for 4 years and decide on a major. What I did is I did a program up in Maine where I was on an island for about 2 weeks, and I had a project. It was kind of like a class at the same time, and that experience kind of solidified to me, oh, this is something I want to do in the future. Maybe not in Maine where it's the water's 65 degrees. So
yeah, that's my nightmare.
So obviously that kind of solidified to me. And then if you're in college and you're trying to get this experience, or you're trying to I want research experience, I want this research experience, feel free to ask. Everyone who you're in class with, especially your professors, they want to talk to you. They love to talk about their science. This is the entire podcast, right?
Right, yeah. Just finding everybody wants to talk about what cool things they're doing.
Exactly. And so feel free to be hey, after class, if they have a minute, say, hey, I want to talk to you about your, whatever you're researching. Can we set up a time to meet? And you just go and you just set up a time. Or like me, I guess I'm a little bit more outgoing and less scared.
Outgoing? What a surprise.
I would just walk up to a professor in their either lab or their office and be like, hey, what do you do? And I would just kind of get a spiel of what they're doing and be like, cool, that's awesome. I know that might be a little bit rude, and but that's kind of what I want to do. If I'm trying to know something, I'm going to go out there and get it. And so that's something that everyone has to consider.
Absolutely. What about Zoe? Where are you looking to go from here, and what advice would you have for people who want to do what you're doing?
So, I feel like I'm pretty fortunate because a lot of people, like Charles just mentioned, come into college not necessarily knowing what they want to do, which is totally normal, totally fine. I have always had a passion for marine biology. I've always loved the ocean, which is interesting for someone growing up in upstate New York a little bit, because I was not really anywhere near an ocean, but I just have always loved it. My 12th birthday present was scuba diving lessons, because that's the minimum age you can go, or at least was. I don't know if it's changed. But that's just something I've always loved. So I always made a point to get myself involved in the opportunities that I could to get closer to marine biology. Growing up, going on different educational trips, raising my money to go on them, and all of that stuff. And so coming into college, I believe I'm actually the youngest in this group. And so the advice that I could give is, if you know you love something, go after it. Like Charles said, get involved when you can. And so this last year, even with COVID and everything, I was a little bit struggling, kind of feeling alone, kind of feeling uninvolved with the college, all of this stuff, because it was just a little bit harder to get involved over Zoom. But we kind of, at College of Charleston, I don't know if this is a common thing, but we kind of get spammed with emails from the college about opportunities. They're really good about letting the students know. If you pay attention, they're really good about letting the students know about what's going on, or what you can get involved in. And so occasionally I would read through them. Sometimes I'd be like, oh, this is too much on top of my classes. But one of the ones that I saw was a kind of research matchmaking session, and they had it on Zoom, which was kind of, maybe not ideal. But I was like, I'm going to go because I just feel like I want to be more involved. I came to college to study this thing that I've loved my whole life. I want to see where it goes, So I just kind of on a whim, I had a little bit of time for it. I just hopped on the Zoom call and it basically connected students who would show up to the Zoom. It kind of connected them with different professors. Specifically, what I've always loved is kind of like the weird creatures of the ocean. So the invertebrates, the weird, like not necessarily, I mean, I love the dolphins and the turtles, but I like the more unique animals, like the invertebrates, like I said. And so I went to a couple, and my mentor that I have now, Dr. Podolsky, he is the head of invertebrate biology at College of Charleston. And so I went to his and just kind of like started talking to him, and he gave me his contact info and I gave him mine, and we kind of just started corresponding after that. And through that, I was able to apply and like gain access to this whole new world of undergraduate research and getting involved in marine biology exactly how I wanted to. And so I applied to this program, and I started being in contact with a professor who could give me access to like learning the science that I wanted to get involved in. And so through that, that's kind of how I got introduced to the REU program and all of that. And as only last year was my freshman year. So as an upcoming sophomore, I feel a little bit like ahead of the game, and it feels good to be, like I said, involved in what I want to be involved in. So I guess my advice is like even if you don't have specific goals in mind, just kind of like dip your feet in the water that you can. You see something that slightly sparks your interest, like there is no downside to just giving it a try or like going after it for a little bit. The worst that can happen is either you discover that you don't like it, which is actually something good in itself.
Still valuable information, 100%.
Because knowing what you don't like is just as important as knowing what you do like. And it also just, it gives you the experience to kind of like keep yourself rolling, you know? Because I know especially last year, I had kind of like dips of feeling like, what am I doing? Like, is this necessarily what I want to do? Or, you know, it's different when you're kind of more like isolated, like we all have been last year. And as a first year at college, that was kind of like a little bit of a hard experience, but it definitely benefited me to just kind of like go after what I thought might interest me, or, you know, like get myself to the point where I could keep myself rolling.
Yeah, absolutely. Totally important to just kind of keep pushing yourself forward and reaching out and finding the opportunities, even when it's like, I have so much work to do, but I need to read this email. And then you're like, oh, that's cool, I should do that. And you know,
Yeah.
it's just really important to be seeking out opportunities like that.
And it's something that you hear, I feel like I heard it so much before I came to college, like talk to your professors and like get involved and stuff. And that's such like kind of, I don't know, overly used advice, but it really is valuable when you like follow it. And like Charles said, honestly, people who are passionate about what they're doing want to talk about it. Any professor that you encounter, anybody who is doing something that they love are so approachable, and way more approachable than one might think. And it might be intimidating at first, but seriously, like the more you just kind of go after it and like get your feet in the water, like it is beneficial in any way.
I love it. That's great advice. What about you, Regan?
I'm not really sure what I want to do in the future. I definitely think I'm going to go to grad school. I love science and I love nature and the world. I want to take care of it as the best as I can. I definitely think I want to go into conservation, but, you know, I'm an upcoming junior. I still have some time. I will be working with Dr. Welch in my toads for a good chunk of time in the future as well. And if I could really give advice to anyone who's, you know, scared and doesn't know, is the whole point about science is we don't know. Like, it's okay not to know because we're asking questions because we don't know the answers to them. That's the whole point. So even if you're scared, if you don't know what's going on, if you have no idea what you want to do with your life like myself, it will be okay because eventually we'll figure it out, you know? Eventually Jo is going to find her aliens and...
Thank you.
And your toads will jump one day.
Yeah, they will jump. They'll grow legs and jump, and we'll find answers that we're looking for. So just don't worry.
That's excellent advice. That if you don't know what you're doing, that means you're in the right place because you're trying to figure things out and grow our body of knowledge. And that's the whole point of science.
Exactly.
I love it.
And Joelle, you already kind of told us what you're hoping to do in the future, but any piece of advice that you have?
Yeah, so I actually have something that I wish that someone told me going into college, and it's kind of a personal story. I came into college wanting to go to medical school, and I was sure that I wanted to be a doctor. Already had my whole life planned out. So the advice I'm going to give is not to settle. I got the opportunity my freshman year, and for time's sake, I'm a junior now. So this is about 2 years ago. I got the opportunity to work in a neuroscience lab at a hospital and work in Alzheimer's. And I worked there for about a year, and it was a great experience, but I realized that I did not want to be a doctor. I did not like blood, humans, stuff like that. So I was really, and I'm someone who I like to have things planned out and know what I'm going to do. And I did not know where I was going. So I remembered in high school, I had a microbiology class, and I really liked it, and it kind of was like my first, science is so cool. So I literally looked up microbiology, College of Charleston, and the first person to come up was Dr. Moshe Rhodes. So I didn't know him, I'd never had him. I was a sophomore, so I really didn't know many people in the biology department. And I said, "Hi, I don't know what I want to do with my life. Can we talk?" And he emailed back almost immediately and said, "Yeah, are you free?" I think it was like Tuesday at 3. And I had a class then, but I decided to skip the class to talk to what would soon be my PI and my mentor. And he said, "What do you like about microbiology?" And I said, "I don't know. I just know I like small things and it's cool and I had a lot of fun." And he goes, "Well, I have a astrobiology project and I think you'd be a great fit for it because you seem passionate about microbiology." And I am. I love microbiology. I did not realize how much I loved research until I started doing something I was passionate about. And so I started working in his lab, and I go there for fun now. He said, "Oh, you can come in maybe once a week just to get the feel for it." I was going in 5 days a week. I have never been so passionate about something. So my advice is don't settle. Don't settle for something that you're going to dread going into, that you're just doing to check off a box for grad school. Do something that you love doing. Do something that you wake up every day and you say, "I can't wait to go in the lab and check to see how pink my archaea are." Do something that you feel lucky doing. So that's my advice.
That's excellent advice. Thank you so much. This has been a wonderful episode. I've really enjoyed getting to know you guys and hear about the kind of research that you're doing and just kind of who you are as people. And it's been a great time. Thank you so much for being on the show.
Thank you.
Thank you.
Thank you for having us.
Thanks for listening to this episode of Conservation Connection.
If you enjoyed our podcast, go ahead and subscribe to make sure you catch every episode that we post.
We'd love to hear from you. So if you want to reach out, go to our website, lastchanceendeavors.com/contact and shoot us an email.
We love questions from our listeners, so if you heard something that you want to know more about, be sure to let us know.
If you've got a minute to spare, leaving a 5-star review on Apple Podcasts will help other conservation-minded people find the show. We'd really appreciate it.
A big thanks to the people working to protect our planet, and a big thanks to you for listening. Don't forget to tune in next week.